Welcome to the lecture on the virtual brain, Inferring Principles of Network Interaction Underlying Cognition. Virtual brain, or TVB, is an open source software. TVB was developed when we recognized that despite of decades of neuroscience research and going increasingly into details, breakthroughs for brain diseases were lacking. We decided to take a step back and to look again at the whole system. And when looking at the whole system, nevertheless, we wanted to account for the details that do matter for a certain function or disease. Thus, the virtual brain works like a caricature of the brain. It captures the details of the brain that do matter to recognize a certain rule or mechanism of a specific brain function or dysfunction. Brain imaging methods like EEG and fMRI record the summed up activity of large numbers of neurons. At this scale, the most appropriate computational neuronal models are the mean field models that describe the lumped up activity of populations of neurons. The cellular level properties are embodied as parameters that average over the population. They act to modify the dynamics of the neuronal populations. And this example of the population model consists of two distinct mean fields, one of the inhibitory and one of the excitatory neurons. The dynamics is determined by local NMDA and GABA synaptic coupling strength parameters. The population model interacts via the reconstructed nerve fiber or structural connectome. The virtual brain links the world of computational neuroscience with the world of brain imaging. This is why with TVB we can run personalized full brain simulations. And I demonstrate you with my colleague Jessica how we use imaging data to construct a personalized brain model. MRI provides the structural and functional constraints for the model. We reconstruct the different surfaces of head, skull, and brain. We reconstruct the nerve fibers from diffusion MRI, and we parcellate the brain and brain regions, taking into account structure and function. Each region is represented by a population model, and the digital twin allows them to simulate for each brain region neuronal activity that can be translated via a forward model to fMRI, EEG, and MEG. Here you see now a simulation of Jessica's brain avatar. We simulate 22 minutes of resting state. To the right, you see the model equations, summed up input currents, and equation 1 and 2 drive the inhibitory and excitatory population. As a result, we get firing rates in equations 3 and 4. And finally, this leads to synaptic gating in equations 5 and 6. The model generates what we can observe on the large scale via non-invasive measurements, that is resting state fMRI. At the same time, it also reproduces the underlying neuronal interactions and processes that we can not simply measure. Thus, the model allows to integrate information of various scales and modalities in a closed hypothesis of the brain. In the present study, we injected EEG source activity time series of the same subject to mimic the input currents that drive a neuronal population, and rather than using noise as is typically done. The excitatory postsynaptic currents are the major sources of the EEG, and therefore we approximate excitatory postsynaptic currents on the basis of EEG source activity. And the simulated fMRI time series are fit to each subject's empirical fMRI time series that were simultaneously acquired with the EEG. This way, we tune the three free model parameters that scale large-scale coupling and local excitatory inputs. Each node of the brain network consists of a population model. Each we derive with three input currents. First, the respective region's aggregated EEG source activity. Second, the global network inputs. And third, local feedback inhibition. The population models are on the large scale coupled by the structural connectome, taking into account the connection strengths between different brain regions. We generate the firing rates and synaptic activity time series that underlie the fMRI predictions. So here we see how, with a single individual virtual brain with fixed parameters, we capture different scales from firing rate modulations during a single 10 Hz brain oscillation, that is a time scale of 100 milliseconds, via local field potential and inhibition excitation balance, to e.g. power modulations and fMRI resting state networks and their dynamics at the time scale of 20 minutes. By simulating resting state functional magnetic resonance images, we capture the underlying hidden biological processes. In the subsequent slides, we will have a closer look at the simulated and experimental features listed here in the summary figure. After parameter fitting and cross-validation, we had the models of 15 individual adults that were able to reproduce their resting state fMRI. We computed grand average waveforms of the model state variables. Grand averages were time-locked to the zero crossings of alpha cycles and our injected EEG source signals to systematically explore the model behavior with respect to the alpha cycle phase. We see here the grand averages of the variables you saw in the equations previously. Synaptic input current components from the network, from local excitation, from feedback inhibition. And here's a summed up input currents that drives the excitatory and inhibitory populations respectively, resulting in population firing rates and synaptic gating. Left and right axis in panel 3 denote the strength of input currents that drives the excitatory and inhibitory populations. The inputs to inhibitory populations are dominated by the excitatory postsynaptic currents, EPSCs. Consequently, firing rates and synaptic gating of inhibitory populations closely follow the excitatory local inputs, the EEG source activity. Inhibitory populations revert the effect of input currents. Therefore, the shape of resulting inhibitory postsynaptic currents is inverted with respect to source activity shape. The amplitude of inhibitory postsynaptic currents, IPSCs, that drive excitatory populations is several times larger than that of the excitatory postsynaptic currents driving the excitatory population. Accordingly, summed up excitatory population input is inverted relative to the alpha cycle. This results in rhythmic inhibition of the firing rates near the peak of the alpha cycle. In conclusion, the fast population activity underlying fMRI resting state predictions show a rhythmic modulation of firing rates on the timescale of individual alpha cycles. An ongoing balancing of excitation by feedback inhibition generates this pulse inhibition of the firing rate. Now on the right, we see this relation of alpha cycle phase and firing as it is in our simulated data. This phase-firing relationship emerges from the model as a consequence of the model fitting derived weights for global and local excitation. At the peak of the alpha cycle, we have minimal firing. On the right, we see a very similar alpha phase relationship in a monkey brain. Invasive monkey recordings have shown that the alpha phase modulates the firing rate in the somatosensory cortex. When a single alpha cycle goes up and here reaches its peak, the firing rate is low. When the alpha cycle goes down, we have a high firing rate. In our simulations, every time we have an excitatory current going in a neuron population, we have an inverse current blocking it out again. In animal recordings, this behavior has been demonstrated as well. This is called excitation-inhibition balance. Here you see the inhibitory activity and the excitatory activity. In our model, as a consequence of parameter fitting, the amplitude of the inhibitory currents is 5 times larger than the amplitude of the excitatory currents. This behavior in our model is the result of fitting to large-scale fMRI. In mice and rat recordings, a similar ratio has been reported. In addition, we find a time delay of circa 4 milliseconds in simulations between excitation and inhibition. This delay has also been reported in invasive recordings. This delay has the effect that firing increase during excitation is limited to a short time window. This increases the temporal precision of neuronal responses, since the neuronal response is self-limiting. Mechanism of excitation-inhibition balance has been associated with various cognitive, mental, and pathophysiological functions. The next step, we explore how our model translates EG alpha oscillations to fMRI. Models were injected with artificial alpha oscillations consisting of 10 Hz sine oscillations that contained a single brief high power burst. The orange envelope of this burst indicates the increase of the alpha power. While positive deflections of the alpha wave generated positive deflections of inhibitory population firing rates, negative deflections were bounded by the physiological constraint of 0 Hz. That is, inhibitory populations rectified input alpha oscillations such that only the positive half wave had an inhibitory downstream effect. As a result, feedback inhibition increased for increasing alpha power, and consequently, the average firing rates, synaptic gating, and ultimately fMRI signals decreases with increasing alpha power, as we find in experimental observations. Also, this behavior corresponds with monkey electrophysiology. And the model simulations, alpha power modulates the firing rate. In monkey recordings, the same relationship has been established. The firing rate is negatively correlated with the alpha power. The same holds true for task performance. The monkey performs better during periods of lower alpha power. We next analyze the relationship between alpha power fluctuations and fMRI oscillations. We generated artificial alpha activity consisting of a 10 Hz sine wave that was amplitude modulated by slow oscillations with cycle frequencies between 0.01 and 0.03 Hz. As in the previous example, inhibitory populations filtered negative alpha deflections during epochs of increased power. We found that increased alpha power leads to increased feedback inhibition of excitatory populations. This introduces a slow modulation of population firing and subsequently synaptic activity and fMRI. This mechanism explains the empirically observed anti-correlation of alpha power and fMRI. In addition, we found that the cycle frequency of the alpha power modulation influences the resulting amplitude of firing rate modulations. Synaptic activity and fMRI. This can be explained as follows. Gradually accumulating excitation augments slower oscillations more than faster oscillations. The large time constant of NMDA-ergic synaptic gating of 100 milliseconds compared to only 10 milliseconds on GABA-ergic transmission leads to accumulation of activity. And this consequently results in the emergence of frequency-amplitude relationships and scale-free fMRI power spectra. So now with a disabled large-scale coupling, the resulting firing rates, synaptic gating, and fMRI waveforms showed no frequency amplitude dependence. When turning off the large-scale network coupling, we see the difference here. In the case of deactivated large-scale coupling, all amplitude peaks are approximately equal. The amplitude modulation in downstream signals disappears. Here we see the power spectral densities, left for the empirical data, middle when large-scale networks is coupled, and right when the large-scale network coupling is turned off. For empirical data and activated large-scale networks, we have a power law exponent around minus 0.8. For deactivated large-scale coupling, we see a flat power spectrum. So here we are looking at the parameter space and in particular the prediction quality for different features in empirical signals, resting state networks, raw fMRI, functional connectivity, and power law beta exponent. The parameter space is spanned by the global coupling and local inhibition values for all brain regions. Colors indicate the average correlations between simulated and empirical data for panels A to C, and the absolute difference between exponents of power law fits for panel D. The dark red that we find in a diagonal in all four panels suggests good prediction quality is converging for all empirical features in the same parameter range. It suggests that biological realistic features emerge at a proper balance of long-range excitation and local inhibition. We now have learned how the brain mechanistically links all these observations together. And here's the hybrid model once more in action. Next, I show you how we can simulate the effect of altered molecular pathways on large-scale brain dynamics. Amyloid beta is a protein that accumulates in Alzheimer's disease. Studies with mice and cell cultures indicate that amyloid beta affects inhibitory interneurons, leads to disinhibition, and increases activity of pyramidal cells. Amyloid beta distributions in the brain can be measured with positron emission tomography, PET. We get a beta amyloid load for each brain region, as you can see here in the colorful brain. We thus can implement the signaling cascade in our model. We introduce a amyloid beta load dependent decrease of the coupling of inhibitory populations to pyramidal cells in our population models. This leads in our simulation to hyperexcitability with increased beta amyloid load. We now can study how large-scale brain dynamics changes when molecular pathways change. Here you see the population model we are using in the study, the Jansen model. It consists of 3 populations: inhibitory interneurons, pyramidal cells in the middle, and excitatory interneurons to the left. We implement an amyloid beta load dependent decrease of the coupling of inhibitory populations to pyramidal cells. This leads in our simulations to hyperexcitability with increasing beta amyloid load. We now can study how large-scale brain dynamics change when molecular pathways change. Here's a bifurcation diagram that shows the possible states of our neuronal population model. Axis Y depicts the postsynaptic potential of pyramidal neurons. Axis Z, the postsynaptic potential of inhibitory interneurons, and the X-axis depicts the excitatory input from other brain regions. When we vary the network input, the behavioral repertoire of the neurons changes. So here you can see how the activity winds around the blue surface and hence generates oscillations. Or here you see just a black line when neuronal activity does not change. So now we vary a second parameter J, the connection strength between inhibitory interneurons and pyramidal cells that is modulated by the regional amyloid beta protein load. In the right panel, we see resulting intracerebral EEG or local field potentials. The manifolds of the model give us a hint on what we can expect, the repertoire of the population. Or the other way around, if we observe different dynamics in empirical data, such as EEG or intracerebral recordings, we can try to infer the parameter changes that in turn indicate underlying biological changes. And in this way, in future, the virtual brain inferred parameters may be used like lab values for clinical decisions. This is the vision. Here we see on the y-axis the EEG frequency and on the x-axis the number of regions where that frequency occurs predominantly. We see the results of simulations of brains of individuals with Alzheimer's disease in red, mild cognitive impairment in green, and healthy controls in blue. The Alzheimer's disease patients had significantly stronger slow rhythms, theta activity in their simulated EEG. This effect is a consequence of including beta amyloid load from PET data in our otherwise identical virtual brains. The slowing occurs in the real experimental data of Alzheimer's disease patients. So here we reveal a possible underlying mechanism that links the metabolic pathway scale with large-scale brain dynamics. Memantine is an NMDA antagonist used in dementia. We modeled NMDA antagonism by virtual memantine. This NMDA antagonistic effect was simulated through a modified coupling from excitatory populations to the pyramidal cells. The y-axis indicates the dominant EEG frequency and simulations of brains from patients with mild cognitive impairment, Alzheimer's disease, and from healthy controls. We see that virtual memantine treatment, indicated by the red dotted line, shifts the Alzheimer's disease group to the level of healthy controls and patients with MCI, mild cognitive impairment. I showed you that we are able to simulate functional connectivity with brain network models. In the final part of this presentation, I demonstrate how we plug neuronal circuits that can simulate decision-making and working memory in the brain network models of 732 individuals from the Human Connectome project. We did this to identify possible performance differences and to infer the underlying neural mechanisms. The decision-making working memory circuits consist of 4 excitatory populations that are mutually inhibiting. 2 populations are sitting in posterior parietal cortex, 2 in the prefrontal cortex. The simulated decision-making experiment is inspired by a random dot motion experiment where subjects have to decide in which direction the majority of the dots is moving. Interestingly, the artificial circuits perform differently well in different virtual brains. If the virtual brains come from persons that score high in fluid intelligence tests, decision-making is better. Thus, those virtual brains of higher scoring people achieve more correct decisions. In this phase portrait, we see the simulated activity for populations A at the x-axis and population B at the y-axis for 4 different brains. The correlation parameter R indicates the strength of functional connectivity in each brain. Average functional connectivity has been shown to increase with increasing fluid intelligence in empirical data. The thin gray lines in each phase diagram represent 1,000 simulations. Orange represents the mean trajectories averaged according to the decision made, that is either for A or for B. The orange trajectory initially runs along the red diagonal. On the diagonal, activity between the 2 competing populations A and B has similar values. When moving away from the diagonal, one population starts increasing its activity more than the other. Once the trajectory departs from the balanced state, due to the cross inhibition of the competing populations, a return to the diagonal becomes difficult. This is indicated by the flow fields, the purple lines in the background. With increasing distance from the diagonal, the flow leads with increasing velocity to the attractors, the black-filled circles. Arriving at an attractor leads to a decision. In brains of persons with higher fluid intelligence, the 2 populations remain balanced for extended time periods. This is caused by the embedding in larger functional connectivity. This gives the system time to average out random noise and for the evidence to accumulate. Evidence accumulation leads eventually to a better decision. In conclusion, the model explains how segregated brain states, when we have no correlation in the surrounding network, lead quite literally to a premature jump into conclusion, while integrated states allow prolonged integration of evidence, resulting in more correct decisions. With this, I would like to come to the end. The virtual brain software and workflows are now available on the European Human Brain Project eBrain's infrastructure.
The Virtual Brain Tool Suite is a large open-source ecosystem with an end-to-end workflow for creating personalized brain network models and simulating them. It is now available on the European Brain Research Infrastructures, or eBrain's cloud. The Virtual Brain on eBrains, or TVB on eBrains, allows users to construct, simulate, and analyze personalized brain network models. Its powerful features can be used from your local computer, making computationally expensive neurological research possible from the comfort of your web browser. The virtual brain integration in eBrains allows users to have an end-to-end experience of personalized brain model creation and multi-scale brain simulation using high-performance computing in the cloud. This makes it possible to process large cohort databases, which in turn makes it possible to produce generalized results. This is a precondition for being able to use these results to develop potential medical treatments, therapies, or diagnostic procedures. The Virtual Brain on eBrains ecosystem software is open source. The software is distributed in the form of execution-ready Docker containers that can be pulled from Docker Hub. The containers can be executed on supercomputers using secure container environments like Saris or Shifter. Jupyter Notebooks enable users to operate the containers on supercomputers directly from their web browser. TVB on eBrains includes a variety of simulation tools, pipelines, and datasets, which we will introduce in the course of this video. The virtual brain ecosystem on eBrains contains the virtual brain simulator that can be used in the cloud with a supercomputing backend, ready-to-use pipelines to build personalized brains that enable users to process their own imaging data or share data with large cohorts, TVB-ready imaging datasets of patients and healthy controls discoverable via eBrains, the TVB multiscale co-simulation web application, which enables co-simulating some brain regions at a finer scale using the NEST simulator, fast and parallelizable hardware-near implementations of TVB models, containerized for direct use on eBrains via Colab notebooks. The INCF training space, with a dedicated area for the virtual brain, where a rich multitude of didactic use cases are available with video tutorials, Jupyter Notebooks, and example datasets. All software and datasets in the virtual brain ecosystem are registered and annotated in the knowledge graph to make them easier to locate and use. The tools are functional and ready to use, and you can be the next user.
Thank you very much for your attention.